Hello. Thank you all for your attention. My name is Shawn Quinn. I am a founder and the Chief Technology Officer at CURIO Genomics. We provide a platform called CURIO, which I'm excited to tell you more about today. Just briefly here to set a little bit of a scope for with the time together, I'm going to be sharing a little bit about Curio, but really we're going to be spending most of the time on a live demo. And so this is very exciting. You never know what's going to happen with live demos, but we're going to try to actually work through a few use cases together. So buckle up, it should be fun. And then at the end, we'll do a little bit of Q&A to wrap up our time together. So first, just a few things about Curio. We are a bioinformatics software company. We do provide some services as well, and we're really focused on accelerating the agrigenomics research use cases. So that tends to focus a lot on plant and animal breeders, but we're really supporting all types of research in the agrigenomic space for those that are trying to develop solutions, find innovations, and get solutions into the market faster to support plant and animal use cases all around the world. A few things that are kind of special about Curio, unique to us. One is that we were built from the ground up for agrigenomics. And so that means things like we were built for handling any types of species, bringing different kinds of species into the platform and really automating that process and the different resources you need to do agrigenomics research. Things like handling polyploidy issues in plants, or like with imputation use cases, being able to build reference panels. So all those kinds of use cases were really built for. Part of the way we're designed is for flexibility. And so you'll see when we get into the demo, you can choose a lot of different things, and the platform can really flex to support a lot of different kinds of use cases. We do that by enabling a lot of well-known algorithms directly within the platform. And then we have a system we call extensions that allows us to extend the platform to support other kinds of algorithms or use cases that are either well-known in the public domain or are proprietary to your organization. One of our specialties is that we do what we call real-time bioinformatics, meaning you don't have to wait on anything within the system. Everything moves really, really fast. And so that is both the way we process data, which I'll talk more about, but also the way we enable visualization of the data. And so when you're interacting with Curio, you're not just looking at text files at a command line, you're actually able to visualize and see what's going on in these genomic data sets. You're allowed to query them in real time, get answers literally in seconds, so that you can make an informed research decision and then move on and ask the next question or feed that data along to other types of systems. And on that point about integration, we're really built for integration as part of a larger enterprise if you need that. And so that includes things like being able to automate pulling data in from sequencing platforms, or being able to push data downstream to other systems that might use, say, genotype information for predictive breeding value programs. And so we're really built for automation and efficiency at scale. I'll talk more about scale here in a minute, but just to note that one of the things that is unique to us is the massive amount of data and the way we process data at scale. That's both for large, like whole genome sequencing data sets where you've got just terabases or even petabases of data, but also when you're processing lots and lots of samples. And so those are the kinds of use cases that people are using Curio for when they really need, massive scale and they need to get to results quickly. That's part of the solution that we're providing. And lastly, I'll just say here that we also have we are a cloud-based compute platform, and the way we provide the platform to our clients is essentially data volume based. But the way we do compute is very different in the way we actually take advantage of cloud resources. That allows us to be incredibly cost efficient when you're processing either large amounts of large samples or really small samples, but you've got lots of samples. We can actually give you predictable and a very cost efficient way to get to the results that you need. One more word here about scale. We've been engineered from the beginning to handle massive scale solutions. My background is actually in software engineering, and many of the people that have worked on Curio have that kind of background. And so that has allowed us to think about bioinformatics a little bit differently. Part of it is how we store data. And so we have a really unique way that we store data on the cloud. Yes, we have industry standard file formats like BAMs and VCFs and FASTQs, but we also take all that data and we have a unique way we store it where it's highly indexed, which allows us to do some things that are kind of amazing in the way that we both compute the data, but also make it available to you for query purposes and visualization purposes. Part of that then leads us to be able to use scale at a really a massive way in the way that we take advantage of thousands and thousands of CPUs on demand to process either single samples all in parallel or many samples all in parallel. And so things move really fast because of the unique parallel architecture of the way the platform works. And then I'll just mention here again, part of what we're trying to do is get this power in your hands. So you can do this all on your own. You don't need us to take advantage of the architecture. It's set up with an intuitive user interface that you can interact with on your own. You don't need to be an expert bioinformatician. You can simply interact with your data sets, and you'll get a sense of how that looks here in a minute as I get into showing you the demo. Lastly here is just to notice where we sit in the workflow. So we are a bioinformatics a platform company, and we're providing the platform to our clients to use on their own, with or without our help. We really are agnostic to the type of sequencer technology you're using or the type of library prep technology you're using, or if you're using capture chemistry or how you're doing your DNA extraction. We have partners that can help serve in these cases. If you need us to help with that, we're happy to point you in the right direction so that we can have a full stack solution. But just note where we fit is that we are really the bioinformatics solution and can support a lot of different kinds of sequencing or library prep technologies. And again, we have partners that can help with that if you need that as well. So that's where we sit in really focused in the NGS data analysis use case. So with that, let me get into a little bit of a demo here. So I'm going to do a few different things. First, I'm just going to give you a little bit of an overview of Curio. So this is Curio. When you log in, you would see something like this, and you'll have access to a different organizational account. I'm in this particular organizational account here we call public, but you would have your own. Within your own account, one of the things that's worth noting is Curio, we don't license Curio by seat licenses, and so you can have as many users as you'd like. And so you simply come in here and you say, I want to add, a different person to my account. You set up what sort of level of privilege you want that user to have, and you click add, and then they will get an email that allows them to authenticate and access the data that you have within the system. Once you get into genomics projects, then you can create different kinds of genomics projects for the different types of analyses or projects you want to do. You give them some type of a name and you create them, and then Curio will give you a space then that you can actually begin dropping data into. So for example, in this project that we created, I could come in here and I could begin uploading data into the system. There's different ways that you can pull data into the platform. You can simply, upload data through your web browser. You can also pull in data from resources online. So this is a common use case for pulling in genomic resources, such as from the SRA or the NCBI, where you want to pull in data that's available in the public domain. And then we also have authenticated systems for connecting data, pulling data in and out of the system. We call those app connectors, where you can register things like S3 buckets or other things that you want to push or pull data out of. And you register those, and then Curio lets you choose those here that you can pull in data directly from those authenticated resources as well. Once you have data in the platform, you can then select it and begin operating on it. So for example, if I wanted to analyze this data, this FASTQ data here, I could say I want to start an analysis on it. And then I have different types of options here that I can choose depending on the type of analysis that I want to run. So if this was DNA data or RNA data, is it short read data? Is it long read data? What kind of data is it? Does it need imputation and so on? I choose the algorithm that I want to, sort of the analysis type that I want to run. I select the data that I want to process. I can do one sample or I can do many samples all in parallel. You can see here CURIO is pairing this data together because it's treating it as paired end read data. I then choose how I want that analysis to run. So CURIO will give you these panels that are unique to the type of analysis that you're asking CURIO to perform. This is an alignment analysis that I'm running, and so I'm going to choose the reference assembly that I want to use. This is dog data, so I'll choose CanFam4. You can see there's a lot of different assemblies that are already built into CURIO, but you can also bring in your own custom reference assemblies. And this is a common use case where people either have their own reference assemblies or they're not yet in the public domain. And so you can simply drop those into the platform, and then CURIO makes them immediately available and knows how to index them and get them ready for you to use fully automatically. You can also like kind of quickly here, choose different bioinformatics options. So you're not having to stitch together a traditional pipeline. You're sort of choosing what you want to do here through the user interface. So I could say, for example, I want to get rid of, low quality data off the end of my reads. I also want to say, maybe I want to downsample to 500,000 reads because I want to run a low pass experiment here. I can choose these different kind of options here in the tool. And then when I click start, Curio will actually then begin doing the work of actually processing that data on the cloud for us. And so, for example, here, this is an example of an analysis that was run, an alignment job here. I can see, how that job was run. It was a BWA alignment analysis. It had these inputs and outputs, and these are the options that we chose when we ran the analysis. This is the version of the software that we ran. And then we can sort of see how this performs. And so I can sort of get a quick sense here of, okay, this analysis, it broke it down into 340 different parts. We spread those parts across the cluster. Each part took no more than 5 or 6 minutes. So it did hundreds of thousands of reads per second. So what would have taken about a day if you had just run that job for this whole genome analysis on the cloud, it would have taken about a day. Cura did it in about 18 minutes because of that massively parallel architecture. And then once we've gotten an analysis in the system, we can then start looking at it. So I could come in here and say, I want to start visualizing. In this case, this is whole genome sequencing alignment data. I want to see like, what does that look like? Okay, so I'm looking around here and I can see I've got, a gene here with some information coming through here. I want to, compare a couple of samples to each other and get a sense of how these samples compare to each other for a couple of different dog samples. So I can see this dog has a SNP here and maybe a heterozygous SNP here that this other dog doesn't. And so I can sort of get a sense of how that works within the system. And I can even track things that I want to collaborate on and say, snip to investigate here. And I can track that as an annotated content that other researchers in my lab can then see. And then I can restore those comments to see like comments that I made in the past. I want to restore and kind of get back to what I was looking at to kind of remember what I was visualizing when I was doing research earlier. So there's a lot of different tools here within the system that you can interact with the visualizers in different ways depending on the type of analysis you want to run on the fly here to get a sense of what's going on with your data at different areas of the genome. So that's the first part, just to kind of get you oriented to what the platform is. Let's talk about a couple of different use cases now. So one is the low pass imputation use case. So this is a common use case that we're doing more of these days where you're starting with low pass NGS data, and then you're trying to predict the genotypes of the plant or the animal using an imputation-based genotyping analysis approach. This works really well when you're trying to get a lot of information in a very, very cost-efficient way. So let me show you a sense of how that works here within the platform. So I'll stay in this project here. So if you want to run an imputation analysis in CURIO, there are a bunch of different tools that CURIO has available. So for example, I can choose here that I want to run an imputation analysis with an algorithm of choice, like I can use Beagle or Glimpse. If I wanted to build my own reference panel, CURIO has in it the tools for doing things like variant assembly filtering and phasing and joint genotyping. It can do that incredibly fast, for hundreds or thousands of samples that you might be processing as part of a reference panel build. Once you've selected some data that you want or the algorithm that you want to use, so I could, for example, here do a low pass imputation experiment here. I can choose the reference panel that I want to use as my imputation source. Here I'm using one that's in the public domain, this dog 10K reference panel. But again, you can also build reference panels directly within Curio for your own novel species. I can then tune these different options of how I want the imputation algorithm to run. When I click start, Curio is then going to kick out a report for me that shows me those imputed results. So let me just show you quickly what this looks like. I'm going to actually show you some high coverage data on this is a particular dog. And if I zoom in here, this is high coverage data that you can kind of get a sense for. I've got these genes down here that are coming through, and I've got these different alleles that are coming through in this particular dog. If I could compare a couple samples to each other and get a sense of, you can see how these two dogs are very different in this area of the genome. They have different genotypes. If I bring in the low pass data for this dog, you can see here, this is the low pass data. I'll zoom in a little bit closer. And I've got some, let me zoom back out here. So I'm in a spot where there's some heterozygous, a little more interesting information here, say down here. So I've got this dog, I've got this low pass data where we have some information coming through here. And then down here at the bottom, we have the high coverage data for that same dog. I then want to look at, so this low pass data here, if I look at where that came from, like this particular allele here, we have an A here. Seen in the low pass data, if I look at where that came from, we have a homozygous A that was called from just these two reads in this low pass data. So we don't have a lot of information here. We've got gaps in the data, right? Because this is low pass data. So that's what is feeding this track of information here. This is then the high coverage data that we're using as a comparison. So now I'm going to take the low pass data and I'm going to use imputation to see what's the predicted genotype of this particular dog using just the low coverage data and a imputed based reference panel. And so you can see how we're getting a really nice picture here of a nearly perfectly concordant picture of the genotype information that was true of this particular dog using only this low pass information. And you can see here it even corrected some things. So like these homozygous allele calls here, it actually corrected them to be heterozygous based on the haplotype information that was available in the reference panel for that particular, that was for all the dogs that were in the reference panel, it was able to correct the genotype of that particular call for this particular dog. So we could bring in a couple of different samples and kind of look at these side by side to get a sense of, I got two different dogs here, different information you can see here for this dog versus this dog. We're again getting really nicely concordant results with very different low pass information feeding into these imputed results. So that's a low pass imputation example here. Here's an example of what I was just showing here, same idea. But here in Curio is highlighting in red the areas of the genome that were imputed, and in green is where the low pass data is coming from. So different ways that you can visualize those results for your imputed based genotype analyses. One more analysis I want to show here is a capture based approach. So this is another approach we're running into a lot where you're trying to transition from something like an array solution, or for the first time trying to build a NGS-based genotyping program for a smaller set of markers, maybe 10,000 to a couple 100,000 markers, potentially to replace an array-based genotyping approach. We've found that with the right chemistry and library prep process and using Curio, we can actually get the cost for genotyping with capture down even below array-based genotyping. So let me show you what that looks like in Curio. So I'm going to jump to a different project here, a cattle project. So this project was actually done with a kit from Twist to capture certain areas of the genome. So I can show you that here if I zoom in. And let me turn on the gene annotations here as well. So we have a gene here. And you can see these, if I zoom in here a little bit closer, you can see we've got these places in the genome where we have these blue bars across the top. Those blue bars represent the capture-based baits, the kit that was used to capture reads. And you can see we're getting a nice stack of reads here right around where we captured. So if I zoom out a little bit again here, you can see, everywhere we have one of these blue bars, we're getting these nice stacks of reads. And then we're getting, allele information here. So I'm getting a nice clear allele coming through in that captured area. I can have Curio look at this data different ways. So I could, for example, look at a quality analysis of that data, see, okay, we're getting really nice quality of our reads coming off the sequencers, nice GC content, pretty low duplication rate. So think the quality of the sequence data looks really good. We can also look at, say, a coverage analysis of that capture chemistry. So here we've got, about, 65% or so of the reads are on target. With any capture-based chemistry, you're going to have some that are off target. But in general, we're getting really nice coverage. This visualization or chart here is showing us that we were able to essentially be all of the positions that we tried to target with about 199.98% of them are covered nicely by our NGS library. And you can see here we're getting around 38x or so coverage for this particular sample. Let me just show you then what the results of that look like. So this particular animal, I'm going to zoom in. This is a particular cattle animal. If I zoom in here a little bit, we can get a sense of what that data looks like. I'm going to actually apply some filters here and say I'm only actually interested in introgenic and coding regions, or I'll say intronic and coding regions here. And I'm going to zoom out here. So we're looking across this whole chromosome here and say I'm only interested in transversions. And I'm going to zero. Oh, sorry, I got the wrong features chosen here. I'm only interested in versions, or excuse me, transversions and maybe these transition mutations. So I can set my filters different ways. I can compare a couple samples to each other. So you can see here we're getting very different pictures of these two different cattle samples. And now I want to compare that result to my actual array data here. So now I'm going to zoom in here a little bit so you can see this a little bit more clearly. We've got two different animals here. The first track here is the NGS-based capture results for the first animal here, and the second animal is in the third track. And then here in the second and the fourth track is the array-based genotype results for that same animal. So here we brought the legacy array data in for these same animals, and we're comparing it to the NGS-based capture results. And you can see we're getting nearly perfect concordance between those two results. I can even have CURIO run a quick concordance analysis for us here and actually measure that for us. And you can see for this particular sample, we're getting about 99.6% concordance accuracy between the NGS-based capture results and the array-based results. When we run that across a bunch of different animals, we found that as long as we had at least 30 nanograms of DNA input, we could get a really nice, we got really nice concordance results across lots of different animals by, again, comparing the NGS to the array-based results. So just to wrap up here quickly, before we go into Q&A, just to share really the key kind of messages about CURIO. One is that we're really focused on a lot of different ag-specific use cases. We're giving you full control. You're the driver's seat. You're able to do this on your own, and everything we do is fully transparent. There's no hidden proprietary algorithms within CURIO. The way we run the data is very unique, but the actual algorithms you could reproduce outside of CURIO. We're set up for collaboration, both within your team, but also within the ecosystem of partners that we work with to provide other pieces of the NGS puzzle. And as a reminder, the way we use cloud resources is very unique because of the way we store the data and also the way we use compute that allows us to be very cost efficient in the way we run analyses on the cloud, but also able to provide to you very predictable cost structures so you know exactly what your costs will be up front. Those are some of the things that really make CURIO unique and that we're able to provide as part of our solution to our partners. So with that, here's some information about how you can find us, and looking forward to digging in and talking a little bit further here during some Q&A together. Thanks.